Hello everybody. Today I will present the study of FLT3 tyrosine kinase inhibitors synergized with BCL-2 inhibition to eliminate FLT3 ITD acute cells through BIM activation. Around 30% of acute myeloid leukemia or AML is underpinned with film-like tyrosine kinase 3 or FLT3 mutation, which is a gain-of-function mutation. This mutation has two types, including all FLT3 internal tandem duplication of FLT3 ITD that is associated with the increased chance of relapse and poor survival. Another type is FLT3 tyrosine kinase low or FLT3-TKI mutation, which is unclear prognosis. This mutation leads to increased downstream signaling pathway and result to uncontrolled proliferation with apoptotic resistance. However, FLT3 is targetable lesion. The invention of tyrosine kinase inhibitor for FLT3 or FLT3-TKI show in vitro and in vivo activity. Unfortunately, a single agent FLT3-TKI for long-term show resistance. The combination strategy for FLT3-TKI to another agents for AML is still required. One of highly interested molecule for AML patients is BCL-2 family member, which is a central regulator for cellular apoptosis. BCL-2 family member categorized into pro-cellular protein and pro-apoptotic protein. For pro-cellular protein, the BCL-2 is overexpression in over 80% for AML patients. And apoptotic protein is included of the apoptotic executioner, BCL-2 homology 3 or BHTLD protein. Looking into the mechanism and the BHTLD protein. From this cartoon here, BHTLD protein can directly activate apoptotic executioner or inhibit the pro-cellular protein. Two of these mechanisms can result to the mitochondrial outer membrane permeabilization and lead to cell apoptosis. From this idea of BHT mechanism, the invention of BH3 mimetic small molecule can be a apoptotic enhancer. And one of interested molecule here is the venetoclax that bind to the BCL-2 or we call BCL-2 inhibitor. So this study aims to study the effect of combination treatment between FLT3-TKI, gilteritinib, and BCL-2 inhibitor venetoclax, both in vitro and in vivo experiment. For material method, for in vitro experiment, the AML cell line and patient sample were provided for investigation. The details for each cell line I will provide from the later slide, and the patient sample include of FLT3 ITD both of de novo and relapse setting, and FLT3 vital as well. The mouse model include of the cell line transplantation, syngeneic mouse model, and patient-derived xenograft. The experiment including of the proliferation assay, apoptotic assay, whole immunoprecipitation with Western blot, and cellular transfection with small difference RNA. Looking to the first experiment of the proliferation assay of cell line and patient sample. The AML cell line include of FLT3 ITD AML, FLT3-TKI resistant FLT3 ITD, and a control which is no response for FLT3-TKI. For the AML cell line, both of FLT3 ITD and TKI resistant FLT3 ITD show that the combo with the combination of gilteritinib and venetoclax together result in extremely reduction of proliferation that significantly decrease compared to the single agent. While the patient sample of both de novo and relapse cases show the similar result in reduction of proliferation compared to single agent. Looking for apoptotic assay. On the left side, show the apoptotic level after treatment for any kind of agents in AML cell line. The gray color bar here is a combo or combination treatment result in higher level of apoptosis compared to single agent and control. And the right side is a patient sample. In similar result, the combo show the higher level of apoptosis in dose-dependent manner. Next, the investigators try to explain the overcoming the resistance mechanism by using venetoclax. From the previous study, report that the rebound reactivation of ERK or MAP kinase signaling in response of treatment FLT3 ITD with FLT3-TKI. They show that at 24 hours, the rebound level of ERK or MAP kinase signaling happened in most of cell line that confer to FLT3-TKI resistant mechanism. Back into this study, the single FLT3-TKI genomics also showed the similar result as the 24-hour for phosphorus or BAP kinase. Interestingly, the venetoclax showed the decreased expression in signaling pathway at 24 hours include of the phosphorus. Much more interest happened in the combo because of in the initial first hour it can dampen the expression of downstream signaling, and also 24 hours much more extremely happen. By this experiment show that the using venetoclax can overcome the resistance mechanism by inhibit MAP kinase. Let's move to explain the apoptotic protein interaction. The Western blot here show the expression of protein in pro-survival and pro-apoptotic protein group, and the combo treatment show the extremely much more decreased MCL1 pro-survival protein reduction compared to other agents, but no different in pro-apoptotic protein expression. Interesting result looking into the co-immunoprecipitation, which is to demonstrate the protein interaction between pro-survival and pro-apoptotic protein together. In the column here is demonstrate the BIM with other protein interaction. After treatment, they found that the genomics can decrease the MCL1 in binding, but increase in BCL-2, while the venetoclax single agent increase in MCL1, but decrease from BCL-2. Interestingly, the combination showed decrease in both of MCL1 and BCL-2 binding, and also provide the increase of BAC, which is the apoptotic executioner in binding pattern. This result that the BIM attached to BAC much more higher and leading to apoptosis. For other BHT or protein, there is no change in binding pattern, and this experiment result that the combo treatment increased apoptosis through BIM. Then they moved to found the BIM effect if they knock out BIM by cellular transfection with small different RNA to knock out BIM, and they confirm Western blot here that the BIM expression is decreased after siRNA cellular transfection, and the siBIM show lack of synergistic effect after knockout compared to control, and also decrease in apoptosis compared to control as well. For the last experiment is about the in vivo of mouse model. They're treating the mouse model of TKI about 3 to 4 weeks and measure the fraction of leukemic cells, include the survival difference of mouse. Interesting, the combo showed a significant decrease in level of leukemic cells in all in vivo models, and also showed a significant prolonged median overall survival compared to treatment. This study showed the synergistic effect of the FLT3-TKI with BCL-2 inhibitor together by overcoming the resistance mechanism. Usually, FLT ITD AML increase in MCL1 expression that confer to venetoclax resistance, and the treatment with FLT3-TKI can decrease the MCL1 expression. Interestingly, the combo can much more decrease the MCL1 expression as shown. And for the FLT3 treating with FLT3-TKI for a long time can result in rebound of phosphorus or MAP kinase signaling that lead to FLT3-TKI resistant mechanism. The venetoclax can decrease or dampen the MAP kinase activation, and the combo also showed much more effect with this one. This study showed the demonstration of mechanism between these two agents and lead to increased cell death, and the BIM is the major load from the apoptotic machinery here. In conclusion, FLT3-TKI together with BCL-2 inhibitor shows synergistic effect for TKI and for FLT3 AML both in vitro and in vivo by inhibiting proliferation, increase apoptosis, and counter resistant mechanism. This study supports the clinical trial in FLT3 mutant AML patients. Thank you for your attention.